All right, so I wanted to welcome everybody to the Simpson Faculty Speaker Series. My name's Chelsea and I am part of the marketing team here at Simpson College. During the presentation, feel free to send me any questions you have for the speaker through the chat feature. And then towards the end of the presentation, I'll go ahead and start asking those questions for you guys. Also, we ask that during the presentation, if you could mute your microphones and your video. And then further, I'd like to introduce our speaker, who is Dr. Pat Singer. She will be presenting Repurposing Drugs to Fight COVID-19. A little bit about Dr. Pat is she is a molecular biologist. She has taught at Simpson from 1982 to 2017. During this time, her and her husband raised 2 children. Her daughter is currently an ER nurse at Mercy One downtown, and her son is a pediatrician in the emergency medicine center at the Children's Hospital in Michigan. And while COVID-19 has altered her retirement plans a little bit, she is hoping to do a little more hiking and volunteer work. She's gotten to do a lot as a grandma during this time. So Pat, I'm gonna go ahead and turn it over to you. If there's anything I missed, feel free to chime in and I will let you get to presenting.
Okay, that sounds good. All right, so I have watched a lot of presentations. Now, wait a minute, I gotta get to me.
Pat, you just muted yourself.
Mute myself. Okay, got it. Can you hear me now?
Yes, we can. Yep.
Okay, got it. Thanks. Thank you. Okay, so I want to say at the outset that I'm not a clinician, so I can't answer any clinical type questions about the drugs that I might be mentioning tonight. I am going to stop about a third of the way through the presentation to see if there's any questions. So send your questions in as we go along. Okay. So there are currently, if you look at if you look at clinicaltrials.gov, there currently are over 2,300 clinical trials related to COVID from all over the world, not just the United States. And about 2/3 of those drugs of those trials involve drugs and seeing how these drugs do with COVID patients. 100% of those drugs being tested are repurposed drugs. Drugs that had had been useful for some other disease or some other condition in people. And there's a good reason for that. If scientists today were to develop a new drug that was could specifically treat COVID, it would take about 10 to 12 years to get an FDA approval on that particular drug. So it really does make sense to explore drugs that already have been used in humans and get them into clinical trials involving COVID. Before we go on, before we really get into the drugs, we need to know a little bit about viruses. Okay, so I'm going to talk about what is a virus and what kind of cells can it infect. And I think I need to do this. Okay, so I think of a virus very much like a flash drive. It is a package of genetic information. Okay, it's not useful genetic information until you plug it into something. So the parts of the virus that are like a flash drive is we got a spike protein, the red structures, and they really aren't red in real life, by the way. And the spike proteins are very much like the plug-in part of the flash drive. There's a protective coat around the information stored in your flash drive, and the same is true for a virus. And the protective coat around the COVID virus is both lipid and protein in nature. And then tucked inside the flash drive and tucked inside the virus is all of the information that needs to be read. So the virus carries genetic information to reproduce. keep the cell alive long enough for it to reproduce. Sometimes the virus will cause the immune system to ignore the virus. So there's a lot of different kinds of things that the virus can carry along with it. But viruses can only infect certain kinds of cells. And it's kind of like a flash drive. So I just got a MacBook Air, and it has 2 USB-C ports. Well, a flash drive doesn't fit into that. Okay, so my computer, even though it has all of the machinery and the software to read the information in the flash drive and to use that information in the flash drive, my computer is not a host cell for a flash drive. Okay. My husband has a Hewlett-Packard PC that has about 3 USB ports for the flash drive. So a Hewlett-Packard laptop computer is a host cell, and you can plug the flash drive in and all of the information can be used. Okay, so I wanted to talk about what then are the host cell requirements for SARS-CoV-2. SARS-CoV-2 is just the virus that causes COVID. So COVID is the disease, SARS-CoV-2 is the virus. And I'll try to use the term COVID virus from here on out. Well, let's see. Just like a flash drive, I think all of us have enough experience with flash drives to know that the plug-in is protected by a cap, and sometimes you have to remove the cap before you can plug the flash drive into the computer, or you have to slide the cover off of the plug-in in order to plug the flash drive into a computer. And actually, the COVID virus is just like that. The spike protein's protected so that it can't bind directly to its host cell. Okay, so the attachment site for COVID, the COVID virus, is this protein. It's ACE2. We'll call it a receptor, although it's not really a receptor to anything except the virus. But first, in order to attach the virus to the ACE2 receptor, you have to activate it, kind of like you got to reveal the spike protein so the attachment can be made. And this protein, TMPRSS2, does the job of activation. And you know, sometimes you have to shake your head about whoever came up with that name. But that's a typical cell biology problem. So TMPRSS2 is a protein cutting enzyme, and it cuts the spike protein into 2 proteins. The S1 is the part of the protein that actually binds to the ACE2 receptor. And the S2 is the stem of the spike protein. And it would be used later on in the infection process, but we're not going to cover the S2's job. So now we know we have to find cells in the body that have both ACE2 receptors, so the virus can dock onto the ACE2 receptor, but we also need cells that have at the same time TMPRSS2, so that protein can activate the spike protein on the virus so it can interact with ACE2. And I have a short list of cells that have both of these proteins on their cell surface, ACE2 and TMPRSS2. Many different kinds of cells in the lung. So the lung tissue gets hit hard by the COVID virus because so many cells will have both of these proteins on the cell surface. Cells that line the nasal passages. Okay, so that could be an entry point for the COVID virus. Even the cells that produce snot can become infected with the COVID virus, and so it's pretty important to wear a face mask. Heart muscle cells, and this has been a problem too. Heart muscle cells have both the ACE2 and TMPRSS2, so they can also become infected with the COVID virus. The lining of blood vessels. I mean, you look at all of the cardiovascular structures that can become infected with the COVID virus, and you kind of wonder, it's no longer like, oh, I wonder why it hits the cardiovascular system so hard. Intestinal cells. So there was some question about can you get the virus from eating things? And salivary gland cells. So it can be in your salivary gland cells. So when you spew out little aerosols of saliva while you're talking, sneezing, whatever, laughing, the virus can be in those aerosols. So it makes sense to wear a mask. Okay, so the big question is though, are there drugs that can block the entry of SARS-CoV-2 into host cells? Well, this is one of these questions that happens in class. Well, of course there is, otherwise I wouldn't mention it. And the drug is a drug called camostat mesylate. So this is a drug that was designed in 2011 by a pharmaceutical company in Japan. And it's been used in humans for 15 years in Japan for the treatment of mostly chronic pancreatitis, but it has been used to treat some post-operative complications. So there's a lot of history of use in humans with camostat mesylate. It is not FDA approved, so it's never really been used in the United States. And there are currently about 4 clinical trials going on throughout the world. The first one started in Odense, Denmark. And what kind of surprised me, and I thought that was kind of cute, the author Hans Christian Andersen lived and wrote in Odense, Denmark. Okay, well, camostat's job is to inactivate the activity of TMPRSS2. So it's like the cap on the spike protein or on your flash drive stays on all the time. So you can never attach that spike protein, you can never attach the flash drive. If you don't cut the spike protein is not available to bind to ACE2 and you don't get good infection. So we don't know any results yet from these tests. So it'll be interesting to see how that works. Okay, so I told you this story sort of to get a lay of the land, is that there are there are drugs that target particular processes in the cell. So I wanted to, now that we've taken a look at one drug, we got a little information about that drug, I wanted to look at, well, how can drugs be used to knock out COVID? Well, just like we saw, we can use drugs that block the entry of the virus into the host cell. Okay, I hope camostat mesylate turns out to be a good drug. It will probably have to be used in combination with others, is my guess. Hydroxychloroquine, and I put down strong maybe. Logic tells us that it would block the viruses getting into the cytoplasm of the cell. And so the virus couldn't complete its infection process. I don't know if that's been proven yet. Okay, and another way to block the internalization of that virus is to use neutralizing antibodies from convalescent patients. And that seems to have been fairly effective. It also seems to have been the treatment of choice for Ebola virus patients. Well, if the virus does happen to get in, then it's possible to block the virus reproduction once it gets into the cell. And this is the job of remdesivir. And I have also seen a lot of clinical trials using leponavir and ritonavir, which are drugs used in treating HIV. And again, there's been a lot of history of these two drugs in HIV patients. And another strategy is just forget about the virus. Let's not even worry. I mean, that virus is eventually going to be cleared from the body. Let's just worry about that cytokine storm. And that's the thing that's making patients who get sick so very, very sick. Okay, so we all react to a virus infection and that response is to involve some inflammation. We need to get cells of the immune system to the site of the virus infection. We need to recruit antibody producing cells and killer T cells and all that kind of stuff. So the cells that are stressed and other immune cells that have come to the scene will secrete protein hormones called cytokines. Cytokines activate immune cells and attract immune cells to the site where the problem is. But it's not uncommon for an immune system to overreact and just go crazy with activation. And so too much cytokine is released. And once too many cytokines are released, it's really difficult to deal with. And maybe some of that permanent damage to the lungs could be the result of the damage caused by cytokine storms. Rheumatoid arthritis is an example of, oh my gosh, the immune system went crazy over this. And so it made sense to use dexamethasone. Okay, and the hesitation to use dexamethasone is you need the immune system to fight the virus, but this immune system's going crazy. And it turns out that the use of dexamethasone is the only drug that can save lives. So if you have COVID and you're on a ventilator, you have about a 40% chance of dying on the ventilator. If you should happen to be given a course of dexamethasone, your chance of dying drops down to 26%. And so that seems like I'd like a better chance, but it certainly is better than no chance. Hydroxychloroquine is also an immunosuppressive type drug. How it works, I don't think people really quite understand yet. Okay, I want to stop here and ask if there are any questions, and then we're going to launch into remdesivir.
It looks like we've had one question come in so far.
Okay.
You say maybe to hydroxychloroquine as a blocker, despite the news reports and studies that say the drug does not show promise. But I wonder about, I'm going to pronounce this wrong, the camostat mesylate. Does one take that as a preventative measure? And if so, what side effects does it have?
Well, the side effects, I guess we're getting into a clinical question that I'm not sure I can answer. Nobody has ever figured out what is the function of that protein cutting enzyme TMPRSS2. And so it appears that you could take it with very few side effects. And my hunch is, and like I said, I'm kind of hesitant about doing anything clinical. If there are side effects, it may be related to the metabolism and the breakdown of that drug so you can clear it from the body. Hydroxychloroquine, it alters the pH of some parts of the cell. And that's not at all clinical. It's the way it works. And I would personally never take a drug that alters the pH of certain parts of the cell, because we're so dependent on those differences in pH for normal cellular function. So for me, even though it might block the movement of the virus into the cytoplasm, I don't care. I would rather rely on these other drugs. So I don't know if I answered that question adequately. If I didn't, ask it again later.
And we have one other question before we keep going.
So now, okay. Is it okay to keep going now?
Oh, sorry. No, I have one more question.
Okay.
Would the cytokine storm suppressive drugs work for an immune compromised patient?
Oh, I don't know. That is a clinical question. I don't know.
Okay. I think that's all the questions I've.
Yeah, okay. Because I don't know how that person has been immunocompromised. So that's a tough one to answer. Okay. So I'm going to move on to remdesivir. Okay, but you're going to need to know a little bit more about biology. Not a lot more, but a little bit more. Okay. One of the things about once you move into the cell gets into the virus, it gets into the host cell. It's just like, well, oh shoot! What if you plug your flash drive into the computer? Now, how are you going to stop it? You don't want to stop the computer from functioning because that would kind of kill the cell. So getting drugs that block viral reproduction once it gets inside the cell is a bit tricky. So we have to find the Achilles heel for these drugs. What are the viruses doing inside the cell that our cells are not doing? Okay, so sorry about a picture of DNA, but I just wanted to remind you that our genome, okay, our set of all the genetic information is stored in a double strand of DNA. And it's fairly easy to replicate this DNA, unless you're in Bio 111 and you have to learn how it's really done, then it's not so fun. But the enzyme that replicates double-stranded DNA is called a DNA-dependent DNA polymerase. DNA-dependent means that this polymerase is reading a DNA template and making DNA. So it reads DNA templates and it makes DNA. And I was going to see if I can stop sharing my screen for just a second and do a little little demonstration on what is a polymerase. Because my granddaughter's learning how to do polymerase activity. Okay, can you guys see me? Okay. So my kids grew up playing with these toys. I don't know if I can even buy these anymore. But here's what a polymerase does. And it's just, to me, one of the marvels of life. All it does is add things to a growing chain. Okay? For DNA, these things are nucleotides. And this happens over and over and over again. Okay, so here's a polymer. DNA is a polymer. And each one of these guys, this is a monomer. Okay, and for DNA, we call it a nucleotide. And we think about all of the living organisms that have ever existed and will exist and exist currently. All DNA polymerase does is to create that genetic diversity is attach beads, attach monomers onto a growing strand. So the whole idea of making polymers in biological systems is just magical. You get so much diversity out of doing one reaction. Okay, so that's my side wonder about this, about cells. Okay, so we have double-stranded DNA that's replicated by a DNA-dependent DNA polymerase. Oops. Now, coronaviruses, instead of having a double-stranded DNA genome, they have a single-stranded RNA genome. That's their Achilles heel. We don't have that. So for them to replicate their single-stranded RNA genome, that's a trick that only coronaviruses can do and other RNA viruses. Coronavirus is not the only RNA virus that we know of. I've given you a small list. I think you've heard of probably all of these viruses. Ebola virus is an RNA virus. Zika virus, influenza virus, it's kind of a funky RNA virus. Hepatitis C virus, measles, mumps, all insect-borne viruses like West Nile virus or Japanese encephalitis virus or Japanese yellow fever virus. Those are all RNA viruses, and HIV. Okay, so we've learned a lot about the COVID virus by knowing something about these viruses. That's helped immensely. Okay, here's the scary part about the biology. Probably in high school, you learned the central dogma of gene expression. What does DNA do? Okay, so you learn that DNA has to be transcribed into a molecule of RNA, and that RNA is read to produce protein. And I really don't care about the RNA to protein. I want to focus on just DNA to RNA. Okay, we have RNA polymerases in our cells. We have DNA polymerases in our cells, but notice both of these polymerases read a DNA template. Okay, so the replicating enzymes are DNA-dependent DNA polymerases. The enzymes that make RNA are DNA-dependent RNA polymerases. Okay, that's what we have, and that's it. Viruses do different processes. So we've already discovered that COVID virus has to be able to copy its RNA to make more virus particles. So RNA-dependent RNA polymerase is used to make new copies of the RNA. We don't have that function. HIV uses its RNA genome to make DNA. Okay, and I'm gonna say we don't have that function either, except it's a very, very slight lie. Okay, yeah, it's a very slight lie. Okay, so we can exploit these enzymes or these tricks that we don't do. And that, like I said, that's the Achilles heel of HIV, of all of the RNA viruses, including HIV and the virus that causes COVID. Okay, I hope that makes sense. I just wanted to point out that viruses have enzymes that we don't have, and those enzymes are certainly great targets for drugs. Okay, here's my kind of crusty self-drawing of an RNA-dependent RNA polymerase, okay? One of the main jobs of this enzyme is to make new RNA strands that can be packaged into new virus particles, and those new virus particles can go on and infect other cells. Okay, so RNA polymerase is just adding bead after bead after bead of nucleotides moving in this direction. Okay, the thing I want to point out is that there are this enzyme has to have two tunnels. One for the template to exit, and another tunnel for the newly synthesized strand to exit. Okay, we don't want these guys we don't want these two pieces of RNA to come back together again. So the enzyme is designed to have two exit tunnels. Okay. Remdesivir is a drug that looks like one of an adenine. So when you need to, when the RNA-dependent RNA polymerase is adding new nucleotides, sometimes it has to add an adenine, and remdesivir becomes a good possibility. And if it does get added, okay, and I'm gonna quit this for a minute. I'm gonna show you what's gonna happen. So we're going to end the screen share for just a second, and we'll go back to our beads. Okay. So what's going to happen is as the template as a template is being read, and we're ready for the next incoming nucleotide, and it has to be an adenine. Okay, the template says put in an adenine. The polymerase has a choice. It can put in a regular adenine, and if it does, it just keeps right on going. Or it can put in remdesivir as its adenine. And it can also keep on going. 1, 2, 3 more nucleotides. Okay, here's remdesivir, 3 more nucleotides. Now we'll see what happens. And after the 3rd nucleotide, remdesivir ends up going through the tunnel and it gets stuck in the tunnel. It can't fit through the tunnel. So that means that the polymerase can't add any more nucleotides because this chain, the newly synthesized chain, can't move anymore. So replication is aborted and there's no more virus particles being produced. Okay, I hope that made sense. Don't freak out at the next slide. That's it for hard stuff. Please do not memorize the structure. It's not going to be on the next test. I wanted to talk a little bit about what does it take to develop a drug, not test a drug, but just to develop a drug. So here is remdesivir. This is what's going through the IV into patients who are getting remdesivir, okay? It has to be able to cross the cell membrane unaided. So that's always a problem for drugs. So Gilead added, pharmaceutical company added this pink part of the drug so that it was, this drug is a little bit soluble in the cell membrane and can cross through the cell membrane. This is not an active drug. We call it a prodrug. Once inside the cell, okay, remdesivir can diffuse into the cell. Once it's inside of the cell, it has to be activated. So it goes through a series of steps inside the cell. Each step requires a different enzyme. And it produces this adenine-looking nucleotide. Okay, and the part I circled here, this blue, this is a cyanide moiety or group. That's the thing that makes remdesivir get stuck in the tunnel. And so that's how it inhibits polymerase. I don't really care if you understand the structure. I love structures, but here's what I want you to know. In developing remdesivir, they had to find a drug that could cross the cell membrane. That's problem number 1. Problem number 2, they had to make sure that the drug could be activated by all of the various enzymes required to activate the enzyme. And then step 3 is then they have to be sure that the drug actually inhibits the RNA-dependent RNA polymerase. So that's all of the development that's going on. Okay, so quick history of remdesivir. Gilead made it in 2009 or something close to it in 2009, thinking, well, maybe it'll stop hepatitis C virus. Didn't work. So in 2015, it dusted the remdesivir off the shelf and tried it for Ebola virus. And it did work, but the antibodies to neutralize the Ebola virus were much more effective. So remdesivir went back on the shelf. In 2020, Gilead got remdesivir back off the shelf and started looking at can it inhibit the RNA polymerase of the COVID virus. And in January of 2020, remdesivir, Gilead got an authorized use, got authorization to use it for compassionate use, which means if the patient is there's nothing more you can do for the patient, and there's no other course of action that you can take for the patient, try remdesivir. In May, I think it was May 1st of 2020, Gilead was given emergency use authorization for remdesivir, which meant if an entire community is at risk, at dire risk, you can use remdesivir. So we've moved beyond the individual to the entire community. And remdesivir has been used since then. It still has not been approved by the FDA. Okay, so how much will it cost? Well, already taxpayers have plugged in $70.5 million in the development and testing of remdesivir. So you've already paid for the drug. A full course of 10 remdesivir administrations cost Gilead about $9.32 to manufacture. And just this week, Gilead announced its pricing of remdesivir. For people on commercial health insurance like United Healthcare or Blue Cross Blue Shield or Aetna, if you're on Medicare, if you're on Medicaid, a 5-day course is going to cost you $3,100, and a 10-day course of treatment costs you $5,700. Some government-funded programs, like the Veterans Administration, is being charged $2,300, and other countries will be paying $2,300 because their governments have negotiated a price for remdesivir. Up until this week, actually, remdesivir has been donated. Gilead's been donating remdesivir. So, okay, that's about all I have. I find drugs extremely fascinating, and I'd like to learn more about their marketing. So that's probably one of my next explorations.